to studying the dynamics of sexual and asexual interactions is that it's a really comprehensive, rich, steeped in natural history system in which they're exploring these sexual, asexual tensions with that rich context around their work. And I could see from her dynamic presentations that she would really go over well in this crowd. So I'm thrilled to have her here. Maurine comes to us from originally from Minnesota, where she did her undergrad at Carleton College before going on to do a PhD at Indiana University in Kurt Lively's lab, the same lab that our own Michelle Seng did her PhD in, and has landed herself now at the University of Iowa, where she's been since 2009, 8, and is continuing to do great things there in her research, but also getting really engaged with outreach work, Darwin Day and Science Boosters Club and that sort of thing. And one of the things she mentioned at dinner last night is that when new recruits, potential recruits, come to her lab, she says that if they're not interested, excited about the opportunities to do science engagement, then it probably isn't such a great fit for them to come to her lab. So it's something her lab really is getting involved in. And so I'm just going to hand over the floor to Maurine, and I'm gonna thank her for giving me a title that really works so well in terms of drawing in the crowd. It helps explain why I get so low attendance for my no sex in the wild talks. I'm going to have to adjust that from here on. So off to you.
And thanks, Jeanette, for the invitation twice. I guess now that I've come here, I've actually made it here. I almost didn't make it here yesterday, but I did. And it's really exciting to be here. I mean, one thing that's fun is I do feel like I'm back in Indiana because both Lauren Reesberg and Michelle Seng were often in attendance at my talks and committee meetings. Well, Michelle wasn't, but Lauren was at Indiana. And so it's fun to be here in such a great dynamic department with lots of really amazing science. So like Jeanette said, I study sex and with a particular focus on natural populations and especially in New Zealand. And I will talk about that work today. I want it for people like Keith and also for Lauren, who convinced me that polyploidy was important when I was in grad school. I do have a more polyploidy-oriented talk that I'm not going to give today, but I'm very happy to talk more about polyploidy with any of you who are interested. So I'm interested in a question that I'm sure you can't have been in a department with people like Sally Otto without knowing that why sex is so common is an important and really long-standing evolutionary question that has really been of interest to evolutionary biologists since evolution was first conceived of as an idea and as a framework. And Darwin, despite his brilliance in figuring out evolution by natural selection, was really stumped by sex. And as he said in 1862 in a paper about primula, sex in flowers, the whole subject is as yet hidden in darkness. Now, as I like to say, in 1862, pretty much everything about biology was hidden in darkness, and especially when it comes to things like genetics. Like, we didn't know about DNA or mechanisms of inheritance, so we have to cut Darwin a break for being stumped by sex. But in about 150 years later, Carl Zimmer says something that's really strikingly similar. While he acknowledges that we figured out the molecular nuts and bolts of sex, we know a lot now about how meiosis and recombination and inheritance work. We still don't know why sex is so common. It remains mysterious. Now, what I would argue that the reason why sex is so common is considered to be problematic by biologists is that it's extremely costly. And what I believe as a biologist who works in natural populations is likely to be often the main problem faced by sexual lineages, populations, is the particular cost of sex posed by the fact that sex in dioecious systems or even in hermaphroditic systems requires either males or allocation to male function. And this is a problem that was first really highlighted and emphasized and documented analytically by John Maynard Smith. And he termed this problem the twofold cost of males. And I'm going to use a simple cartoon to illustrate the nature of the problem. I'm going to be using these are snails, and I'm using classic pink and blue color coding. I apologize for that, but it's easy. So females are pink, males are blue. These two females, sexual and asexual, are identical in every way, except for the manner in which they produce offspring. So this asexual female makes daughters that are genetically identical to herself. The sexual female is going to make both daughters and sons, with each daughters and sons only containing half of her genome, with the other half coming from a male. Now these females are otherwise equal. So after one generation of reproduction, each of these females has produced 2 offspring. The asexual female has made 2 daughters. The sexual female has also made 2 offspring, but she, like most females in systems with both males and females, is going to invest half of her reproductive allocation in sons, meaning that she's only going to produce 1 daughter for every 2 produced by the asexual female. This process will repeat itself generation after generation. Only females are capable of directly producing offspring, so I'm only showing offspring production by the females. What this means is that if we were then to go into this population after only a few dozen generations and count up the number of females in the population who are asexual in red versus sexual in blue, and I've got numbers of females on the y-axis, we're going to see many more females in the population that are asexual than sexual. And again, because only females can directly produce offspring, the number of females in the population are really going to determine the rate at which these populations grow. So this very dire, or this very simple discrepancy in the rate of daughter production between sexuals and asexuals translates into very serious implications for a sexual population, again shown here in blue, invaded by an asexual mutant and her descendant in red, such that after only a few dozen generations, what we expect to see is that this sexual population is going to be driven to extinction by competition with the asexual female and her descendants. And of course, 45 generations is really nothing in evolutionary time, and is what underpins the theoretical expectation that sex should be rare or even non-existent in natural populations. And of course, we know that this expectation is absolutely not met because all or virtually all eukaryotic lineages use sex at least some of the time. So I've told you these 2 pieces of information. I've told you that sex is common, sex is costly. What this then has to mean is that sex has to be associated with, directly or indirectly, some very profound benefits. Now, evolutionary biologists who study sex tend to focus on the fact that sex, to presumably unique extent, is associated with the rapid production of genetic and phenotypic variation, something that asexual lineages are not nearly so good at doing. And this is the reason for why evolutionary biologists who study sex, including myself, tend to focus on causal benefits of sex that are associated with the production of genetically variable offspring. So direct or indirect benefits that have to do with the fact that these sexuals can make genetically variable offspring. The extent to which these benefits of sex explain its predominance in nature still remain an open question, and it's really a main focus of a lot of the work that I do today. What I like to say is the other side of the coin, however, is that there's also a lot of ways in which sexual and asexual organisms are often different besides the way in which they produce offspring. And this is a point that Loren Riesberg, in all of his wisdom, would bring up at my committee meetings again and again between 1999 and 2004, and I ignored him every time. So to my peril, as it turns out, again, I'm not going to talk too much about this, but there is also, again, there's many ways that sexuals and asexuals are different. It turns out that one of these ways in which sexuals and asexuals are often different is ploidy level, with a lot of asexual animals and plants being polyploid. And I've just shown a few examples here. These are our snails. I should acknowledge that this is much larger than life size. You probably figured that out by now. But you know, asexual plants like dandelions and a lot of asexual organisms, daphnia, salamanders, stick insects, leaf beetles, and a lot of asexual fish are triploid or even higher. And as again, I'm sure I don't have to convince this crowd, polyploidy confers a lot of really interesting and still not well understood genomic and phenotypic consequences. So by understanding and characterizing these genomic and phenotypic consequences of polyploidy, we can both learn more potentially about the maintenance of sex and also address the even broader question surrounding evolution of genome architecture and why diploidy is so common in nature. All right, so I'm going to take a step back and remind you again that I'm interested in testing hypotheses for sex. What I would argue that these are really conceptually simple to do, and they're practically actually very challenging, and especially in natural populations. The reason for this is that I would argue that direct tests really require otherwise similar, naturally coexisting obligate sexuals and obligate asexuals. This turns out to be a scenario that's quite rare in natural populations. And it's really non-existent amongst our classic model systems. And these reasons are why I've been really focusing my career on using a really incredibly difficult snail system to use to approach these questions about sex. Like, there's all sorts of reasons why nobody should ever use these snails to study anything. And this is why Michelle Tseng probably did her PhD on mosquitoes in Kurt Lively's lab instead of on snails. But I did snails, and there are some reasons that they actually are useful to apply to questions for sex. So these are the snails in their natural habitat in New Zealand. This is a rocky lake bottom. This rock is probably about the size of my fist, and I've circled these really tiny snails here in red. So they're always smaller than people expect snails to be. So all of you guys have some sort of image of snails in your mind that's like 10 times bigger than these snails. So they're about 4 to 6 millimeters in size, and the reason why these otherwise really pretty nondescript snails are thought to be, or are so good for studying sex, is that to a very unusual extent among natural animal and plant populations, we do see frequent coexistence between obligately sexual individuals, and I'm again going to use this red-blue color coding throughout my talk for sex and asex, and obligately asexual individuals, which are nearly always female. This is a map of New Zealand, and each of these pie charts represents a sample of snails taken from a natural freshwater lake or stream population. And the extent to which the pie is blue versus red represents the extent to which the sample is sexual or asexual. And what these kinds of surveys done by Kurt Lively, and as well as myself and others have shown, is that we both see very frequent coexistence between sexuals and asexuals within the same lake. And this sort of scenario provides very powerful direct comparisons between sexuals and asexuals that seem to be pretty much otherwise similar aside from those ploidy differences. We also see that some lakes are consistently much more asexual than others, like these lakes here, all red. They tend to be asexual year to year. Other lakes, like these mostly blue lakes here or up here, tend to be sexual. So we can compare lake populations with more sexuals to those with fewer sexuals in order to address questions regarding differences in environmental or ecological conditions among lakes that might also help contribute to the maintenance of sex. John Maynard Smith was aware of these snails when he wrote his seminal book on the evolution of sex in 1978, and he actually highlighted the snails. By the way, I call them potamos, so I'll use this language for the rest of this talk. He highlighted potamos as being very promising, saying further investigation of these cases and meeting potamos could be most interesting. And so it was this kind of setup that really got Kurt Lively excited about the snails in the 70s and 80s, and the work that I and others are continuing today. So among some of the other work that John Maynard Smith inspired with these snails was work that I've been doing, which is looking more closely into the ploidy variation in these snails. So we knew starting, I think, in the 1990s that these snails, the asexuals, are indeed polyploid, and they were assumed to be triploid. But I started looking a little bit more deeply into the ploidy variation in the asexual snails, and as I think is often common in natural populations, when you start looking at ploidy variation across a wider sample and across more individuals, I think we often find that there's more ploidy variation apparent than is initially assumed to exist. And this is exactly what we found. We just essentially did flow cytometry of thousands of snails from a bunch of different natural populations. Now I'm adding this color coding of tetraploids in yellow, triploid asexuals in red, again diploids in blue. And what this work revealed is that we actually have ploidy variation within the asexuals as well. And again, we have some consistency from year to year in these ploidy variations within lakes, such that some lakes are consistently high in tetraploids relative to triploids, like this lake here or this lake here, while other lakes, even though they're still all asexual, they're all or nearly all triploid, like these lakes here, this lake in here. So this situation was really nice for us because it enabled us to, at least in part, decouple the effects of reproductive mode variation, so sex and asex, from those of ploidy, by comparing triploid and tetraploid asexuals, and any differences between triploid and tetraploid asexuals could then potentially be used to inform differences between diploid sexuals and polyploid asexuals. Other work that we've done has demonstrated that there are multiple and distinct transitions to asexuality from the ancestral sexual state in the snails. And what this phylogeny here represents are whole genome SNPs taken from the ongoing genome assembly projects for 26 different snail lineages in our species, along with an outgroup, Potamopyrgus estuarinus. This is also a sexual species. And what we can see here is that when we look at the number of transitions from sex to asex that are required to even explain this tree, we see at least 7 independent transitions. we know that there are many more transitions even that are happening within lake populations. So what this indicates is that we have a number of different separately derived asexual lineages that we can then use for a number of powerful comparisons, such that each of these distinct asexual lineages can be considered to provide an independent natural experiment in what it means to be asexual or polyploid. We see the same sort of patterns of independent ploidy elevations from triploid to tetraploid within the asexual snails as well. So for these reasons, a snail system that in a lot of ways is quite intractable does provide some really nice advantages to perform powerful and direct tests of advantages and disadvantages associated with sex in natural populations. My lab group and I then use these snails to address questions regarding causes and consequences, as well as maintenance of sex in these snails, and somewhat more recently, ploidy variation, using a lot of different approaches. We do everything from genomics and genetics through organismal ecology, even some ecosystem ecology, and we do behavior. We pretty much do everything that is of relevance to addressing what I think is a really integrative and broad problem of the evolution of sex, and then related questions involving evolution of genome architecture. So what I'll be focusing on mostly today is the resources made available by a project that we call Polymomics. This is one of my most, I think, clever moments is coming up at this Genome Name project. This is a group of 9 people led by myself, along with my 2 co-PIs. This is John Logsdon, who I think some of you know, Jeff Bohr. And what I'm most proud of is that this whole genome project really is driven by grad students. So every one of these other 7 people here are graduate students who have spent some time in my lab, either as a technician or as a student. And they have been the people to figure out how to do everything from start to finish, from DNA extraction from a very mucousy organism, which as some of you know is non-trivial, to doing the library generation and sequence or genome assembly and annotation. So this again is a project that grad students have done start to finish. And right now we're at the point where we have a pretty high quality reference genome that we have been assembling from this particular sexual lineage here, that's kind of right in the middle of the phylogeny. And we also generated resequencing data from 25 additional lineages from 16 natural populations. So we've done the best we could to achieve the breadth of sampling that you need to really, ideally, do a lot of comparisons both within and across these natural populations. We're also in the middle of generating a genome assembly from this outgroup species, Podomopergus esterinus. Now, we are getting to a point where we have a nice genome assembly, and we have what I think of as might be the most diverse set of data in a genome assembly that I know of to date. We started out with Illumina data. It gave us a pretty crappy assembly. We added kind of early PacBio. It didn't do as much as we hoped. We added 10x Genomics. It in some ways made things worse. And then we did Dovetail Genomics, and again, it didn't do that much good for us. Right around this point, though, we started asking ourselves, how is it that we can have 100x coverage on this genome, or what we think should be 100x coverage, and have such a crappy assembly? We thought we were doing a pretty good job with doing some novel things with joint and hybrid assemblies. And what we realized, and I'm not going to get into this more today, but I'm happy to talk about it with any of you, we actually started realizing that there was a pattern of data of essentially what looked like identical genomic sequences on different scaffolds that were causing this assembly to be more fragmented than it should have been. And so we've been realizing very recently that we actually have a genome duplication somewhere in here, essentially, such that even what we call diploids are degenerate tetraploids. So this has generated a situation where we need very long-range, very high-quality sequencing, PacBio sequencing in particular, to really resolve this genome assembly and give us a lower number of scaffolds and increase our N50 to something where we can really do things with like synteny and so forth. So we're in the process of generating those data. That said, we still have a high enough quality assembly that at this point, the complement of complete and conserved single copy genes does seem to be the best available among current molluscan genome assemblies. So we're able to use this genome to do the kind of specific gene-focused analyses that I'll be talking more about today. So in particular, what we're using these data right now to address are particular expectations of key hypotheses for sex, looking at both harmful mutation accumulation, and I'm going to talk today about harmful mutation accumulation from the perspective of single point mutations, transposable element accumulation, as well as what's going on in genes that are thought to be specific to sex or specific to maleness. I'm also going to be talking a little bit about expectations regarding the type, magnitude, or efficacy of selection in sexuals and asexuals, and with a particular focus on purifying selection and relaxed selection. We're also doing some work with adaptive evolution, but I'm not going to talk about that today. So what I'm going to do now is go further into the work of 3 of the graduate students who are part of the project and give essentially a vignette that summarizes what they're doing, addressing some of these really critical and really, in many ways, untested hypotheses for sex in the context of natural populations. So I'm going to start out with the work of Joel Scarborough. So Joel was a PhD student with me who now finished about a year ago. He's now a postdoc at Colorado State with Dan Sloan doing some really nice mitonuclear coevolution kinds of work that's really building on the work he did in my lab. And what Joel did was use mitochondrial genomes to address questions regarding the extent to which mutation accumulation in asexual lineages is actually likely to be harmful in a way that could influence phenotype in meaningful ways. So, as I'm sure you're all well aware, there's a number of hypotheses that focus on this idea that sex should be associated with more efficient removal of harmful mutations from genomes. And I'm just going to talk about what I think is the most general explanation for this effect, or this sort of consequence of sex. This is the Hill-Robertson effect. I'm going to give a very simple cartoon illustration of how this should work. So what we've got here are 2 chromosomes, black bars. We've got mutations relative to ancestral alleles in C foam green, little b. We've got a good mutation here in yellow, big A, bad mutation, big B in red. So we know that linkage, and by linkage I mean both physical linkage, such that loci are stuck together on the same chromosome, or effective linkage, which is what happens when different chromosomes are always co-transmitted, like in an asexual organism. This should result in a scenario where the extent to which these different sites, A and B, can be selected independently. This is not going to happen very effectively when we have linkage. And this is called the Hill-Robertson effect. And this is expected to lead to mutation accumulation when you have linkage. So what sex does, and in particular recombination, is that it breaks down these linkage groups, allowing loci to be selected independently of one another. So this is at least one reason for the expectation that sex should enable more effective removal of harmful alleles from populations. Now, we have pretty good evidence that mutations that are thought to be harmful do accumulate at higher rates in asexual lineages, including some data that we generated from mitochondrial genomes in Potamos now 7 or 8 years ago. And this is a figure from this paper demonstrating the accelerated accumulation of non-synonymous mutations in the mitochondrial genomes of asexual lineages in red relative to sexual lineages in blue. So this sort of pattern has been documented in many different types of organisms, plants and animals, as well as, I believe, some microbial lineages. So we know pretty well that mutations that are associated with changes in amino acid sequence are accumulating at higher rates in asexual lineages. So what we really don't know much about is the extent to which these mutations that are thought to be harmful, in fact, are having effects on phenotypes of organisms in ways that could contribute to understanding the maintenance of sex or the success of asexual lineages. So these kinds of things are happening. This is as predicted by the Hill-Robertson effect. This isn't that surprising. We showed this accelerated mutation accumulation in the mitochondrial genome of asexual organisms in these Potamos relative to sexual counterparts. This is as expected under Hill-Robertson because in an asexual lineage we have co-transmission, so effective linkage of nuclear and mitochondrial genomes. So what we don't know is whether these mitochondrial mutations are harmful. And this linking genotype to phenotype in this context is not easy. And I'm going to tell you right now that we haven't done it. We don't know definitively whether these mitochondrial mutations are actually harming phenotype associated with mitochondrial function in these asexual snails. But what Joel has done is taken key steps towards addressing this question. He's still working on it, but I think he's made some real progress. And so what Joel did is use patterns of protein evolution to evaluate harmful mutation accumulation in these snails. So he built on this data set that I'd already generated showing accelerated non-synonymous mutational divergence in asexual lineages, added some additional mitochondrial genomes, and then used these data to address the extent to which various types of non-synonymous mutations were actually likely to be harmful. And then he addressed whether these types of amino acid changes that seem to be harmful to a different extent were accumulating in different ways in sexuals and asexuals. So the way that Joel addressed this question was by coming up with a way to categorize these non-synonymous mutational changes in ways that would make reasonable predictions about the extent to which they were likely to influence proteins in meaningful ways. So in particular, Joel took advantage of the fact that we know something about the biochemical properties of amino acids, and that there are these existing classification systems for amino acids with respect to biochemical properties to sort amino acids into categories that essentially are differentiated based on whether the amino acid change is going to be one that will have a big effect on biochemical properties or not. So this is an example of one of the 7 classification schemes that Joel used. This is one based on charge and polarity. As you can see here, we have 4 classification systems that sort amino acids based on whether they are polar or not and charged or not, or how they are charged. And a change from one amino acid to another within a particular biochemical class is a conservative mutation because it's not likely to change the biochemical properties of the protein. This is contrasted to what we call radical change, where we are changing biochemical properties of a protein because we're switching an amino acid, in this case that's nonpolar and uncharged, for an amino acid that's positively charged. Now you can sort amino acids in a lot of different ways, so Joel picked 7 of what seemed like the most orthogonal and the most important ways of sorting amino acids to come up with a kind of mean conservative versus radical score for all of the amino acid changes that we saw within these mitochondrial genomes. He then addressed the extent to which these different types of amino acid changes were accumulating differently from one another relative to synonymous changes and to non-synonymous changes as a whole. So what I'm showing you here, first of all, is the rate of divergence of these different types of substitutions on the y-axis. Here we've got synonymous changes, which provide a neutral or close to neutral expectation for the way that mitochondrial mutations should accumulate relative to non-synonymous mutations as a whole. So what we find is that, as expected, we see a much higher rate of accumulation of synonymous mitochondrial mutations. Not surprising when we imagine that, or when we assume that these are not changing amino acid sequence, so they're not changing protein structure at all, versus the amino acid changing non-synonymous mutations. But then, when we look at conservative and radical mutations, so when we subdivide this non-synonymous class into these 2 different subclasses, we find that there's actually significantly different rates of accumulation of these 2 different subclasses relative to non-synonymous mutations as a whole. Now, these are all at such low rates that we can't see anything on this axis because the synonymous changes are accumulating at so much of a higher rate. But when we zoom in on this x-axis and we look again at the rate of divergence of these 3 different classes of non-synonymous changes, what we find is that, indeed, the conservative changes are accumulating at about a 50% higher rate than the non-synonymous changes as a whole, and more than 3 times as high of a rate as the radical non-synonymous changes. So this tells us that, indeed, the radical changes really are likely to be much more harmful than the conservative changes. They're being removed by natural selection at a higher rate, so they're accumulating at a lower rate. This also tells us that lumping all non-synonymous changes together as a single monolithic class really means that we're losing some information. So we use this information then to assume that radical changes are likely to be more harmful than conservative changes, and then to go through and then address the other question of interest, which is whether these different kinds of changes are accumulating at different rates in different patterns in sexuals and asexuals. So what Joel found was that when we compare conservative changes to one another here on the left, scaled by the synonymous substitution rate, so he's accounting for effects of mutation rate, what he sees is that there's no difference in the rate of substitution of these conservative changes in sexuals and asexuals. You can also see this here from this tree, that we don't see any consistent differences in branch lengths between the red asexuals and the blue sexuals. We see a different pattern when we look at the radical changes, again scaled by the synonymous changes, where we do see a significantly higher rate of accumulation of the radical changes in the asexuals relative to the sexuals. And we can see this pattern as well in the longer branch lengths of the red branches of the asexuals relative to the sexuals. So if we put all these data together, they do point to a scenario where we do see a higher rate of accumulation of harmful changes in asexual relative to sexual lineages. And this is via the fact that we see these radical changes accumulating at a higher rate than in the sexuals. What we still don't know, and what we're still working on, is the extent to which these different patterns of mutation accumulation in sexuals versus asexuals are influencing the function and performance of mitochondria themselves and mitochondrial relevant phenotypes. So what Joel has done is he's adapted a bunch of assays of mitochondrial performance that have been designed for model systems to our snails. So he's done things like respirometry, and he can also assay the extent to which mitochondria are good or not at pulling protons in across the gradient. And we're now doing functional assays of mitochondrial performance in sexuals and asexuals. So I think that we're partway down the road of assessing the extent to which mutations are actually causing discernible and relevant effects on phenotype in sexuals and asexuals in this context of natural populations. So that's where Joel is right now. I'm now going to switch gears some extent to still talking about harmful mutations, but now from the perspective of a different kind of mutation, and these are transposable elements. And this is work that's being led by a current grad student, Kyle McElroy, in my lab at the University of Iowa. So transposable elements are DNA sequences that can change location and create new copies in the genome. And this is famously was described by Barbara McClintock in corn. So because I'm from Iowa, we always like the chance to show corn because we have a lot of corn in Iowa. So this is my Iowa corn. These genes can move, so we think of them as jumping genes. They're also thought to likely, at least much of the time, be parasitic on the genome and often potentially hazardous to the host. Though it's also important to note that there is a really growing appreciation for the potential, really important as well as complex, not necessarily always harmful role for transposable elements in genomes and evolution, with a really nice example coming from this very cool study demonstrating that the industrial melanism mutation in British peppered moths was actually a transposable element. So TEs can sometimes be bad, but maybe not always, and they might provide some nice raw material for adaptive evolution. And we also know from surveys of genomes, as we get better and better at studying genomes, we have been learning more and more about the really major contribution of TEs to some of the most striking and interesting genome variation that we see. So this figure shows the extent to which we see transposon content in the genomes of important model or well-characterized animals like frogs. These are cats. We go from 75% in frogs, 55% cats, 15% in flies, and 3% in this blobby thing that I, again, forgot to look up what it was. But I think that they're either C. elegans or bacteria, but I don't know. But they're small. So we see wide variation in transposon content in different organismal lineages. And we know that transposons in their proliferation are responsible for a lot of the really interesting differences in genome size and structure that we see. So a really good way to understand genomes and a really important way to understand in particular the dynamics of TEs in genomes is to compare TE evolution across reproductive modes and ploidy levels. And I'm going to talk a little bit more about why we can leverage reproductive mode and ploidy variation to study TE evolutionary dynamics. So of course, because TEs are generally important to genomes, it's also going to provide a deeper understanding of what it means to be asexual and what it means to be polyploid. And I'm going to now give some cartoon examples of how sex and asex and ploidy elevation should influence TE evolutionary dynamics and accumulation. So here, again, I'm comparing an asexual to a sexual scenario. These circles represent individuals. These are diploid individuals. They've got 2 copies of 1 chromosome. This aqua box represents a transposable element. So we're starting out here in my cartoon exactly the same in the asexual and sexual population. 1 individual is heterozygous for a transposable element. Now, we typically assume that we need sex to make these transposable elements spread across lineages. So what we see is that the asexual lineage, the transposable element, stays confined to 1 particular lineage, while in the sexual population, this transposable element can spread in the population rapidly via sex from 1 line of descent to another. So the expectation is that pretty quickly we're going to end up with a sexual population that's much more burdened with transposable elements than others. In the asexual population, either via selection against transposable elements or drift, we might even lose the transposable element and end up with a transposable element-free population. So the expectation then is that sex should facilitate a cross-lineage spread of TEs. This, of course, though, is complicated by the expectation that if these TEs are harmful, we do expect that through the Hill-Robertson effect, that sex is going to increase the ability of selection to remove TEs from a lineage. So we might actually, on the other hand, see reduced TE proliferation in sexuals if this particular increase of the efficacy of selection is especially important. Now, this situation gets even more complicated when we remember that a lot of asexual lineages are polyploid. So all else being equal, polyploids should have more sites where TEs can insert neutrally, especially if TEs express any effects that they have in a recessive manner. So here I'm comparing a diploid lineage here. Every red individual has a TE. Let's imagine that being homozygous for a TE is bad. So we might see selection, effective selection against diploid individuals that are homozygous for these TEs, and we can see effective selection purging transposable elements from a diploid population. This process is not going to happen as effectively in a tetraploid population because we don't see selection against these elements until we have homozygosity of those elements in a tetraploid setting. So we can see the much higher, we can see a higher rate of accumulation and a higher transposable element burden in these polyploid populations. And again, this is an expectation that really hasn't been particularly, it hasn't really been tested much at all to the extent that I know in many real organisms. So these questions about how sex and asex and ploidy elevation will influence transposable element evolution really remain to a large extent unevaluated. So I'm going to walk through some very simple predictions for what we think are the most plausible ways in which reproductive mode variation and ploidy variation and their interaction could influence the dynamics of transposable elements. So here I've got sex and asex using the color coding that I've been using to date, diploid sexuals that are blue, red triploid asexuals, and yellow tetraploid asexuals. And I'm going to be using these 3 pluses to represent the highest transposable element abundance. 2 pluses in green are intermediate transposable element abundance, and a yellow plus in yellow, or a plus in yellow is the lowest transposable element abundance. So if we see a pattern when we compare transposable element loads across sexuals and asexuals of these different ploidy levels, if we see a scenario where the highest ploidy levels are associated with the highest transposable element abundance, we might then interpret this as a situation where having a higher ploidy level, perhaps through mutational shielding, is increasing transposable element accumulation. If instead we see a situation where asexuality, but not ploidy elevation, is associated with transposable element proliferation, we can interpret this situation as one where we have reduced efficacy of selection in asexuals against harmful mutations, leading to transposable element accumulation. And importantly, we have this nice comparison between triploids and tetraploids that allows us to rule out a ploidy effect. Finally, a situation where we have much higher transposable element abundance in sexuals relative to asexuals does suggest that asexual reproduction has a positive effect on restricting TE accumulation and that ploidy is not so important. And there's, of course, other possible outcomes too, but these, again, are those that are both the most straightforward to interpret and we think potentially the most plausible in terms of what theory suggests. So what Kyle is doing is he is using our genome assemblies, and he's made a TE catalog to categorize the abundance of TEs, and as our assembly gets better, he'll actually look at the location of these transposable elements across our genomes. And what he's been doing for now is taking these different genomic data sets from different snails of different ploidy levels and different reproductive modes, and he's been figuring out which transposable elements stand out in terms of having especially high or low proliferation in a particular type of snail. So here I'm showing you an example where we are comparing the copy number or the RPKM of particular elements in a sexual snail. This is a blue sexual snail from Lake Maparika. And we are comparing this snail to a triploid asexual snail from the exact same lake. Now we know from all of our genomic and phylogenetic analyses that asexuals are derived from sexuals in the same lake. So it's very likely that this asexual lineage has a recent sexual ancestor closely related to this sexual snail here. And if we plot the abundance of particular distinct elements in the sexual diploid versus the asexual triploid, we see generally pretty good correspondence along this 1-to-1 line here. Notice that this is a log-log scale. We do see a cluster of elements up here that are occurring in especially high frequency in an asexual triploid context. Kyle went ahead and did these sort of pairwise comparisons across pretty much all of our genomic data sets that we have. And he kept noticing the same clustering of very high abundance of these particular elements in asexual triploids relative to the sexual diploids. He noticed, interestingly, the same patterns in asexual tetraploids relative to asexual triploids. So Kyle has been really focusing more intently on these particular elements that are proliferating at especially high rates in asexuals to understand more about what these elements are and what they might be doing. So what this figure is showing you here is actually, this is not a TE. This is a repetitive element with a histone domain, but it came out in our analyses that were focused on repetitive elements because it's repetitive even though it's not actually a transposable element. So here we're looking at this repetitive sequence with a histone domain. This cartoon shows what this gene, this histone array can look like. We've got H2A, H2B, H3, and H4. Here we've got RPKM of these histone elements in our snails. This is a phylogeny, whole genome data from our 26 lineages, or our 20, I don't have the outgroup here, using the same color coding as before, diploid blue sexual, triploid red asexual, and yellow is tetraploid asexual. And I don't probably have to tell you, you can just look, but I'm also going to show you that we see much higher rate of proliferation of these histone elements in asexual triploids here in red and blue, asexual tetraploids in yellow, and across all asexuals pooled in orange relative to the sexual diploids in blue with respect to RPKM on the y-axis. So this is a really striking pattern. We can show that it holds up to, when we do phylogenetic independent contrast, this pattern really holds up very nicely. So we're seeing a very high rate of proliferation of these histone elements. Really intriguingly, we did the same analysis looking at a ribosomal DNA element that also was showing up in that cluster of very highly represented elements in asexuals. This is our DNA element here. We see the exact same patterns with a much higher rate or much higher abundance of these elements in asexual triploids, tetraploids, and asexuals together relative to sexuals. Now, we don't yet have a good enough genome assembly to know whether these elements are occurring actually together genomically in the same place. We do know that when we plot the RPKM of the histones on the x-axis relative to the RPKMs of the rDNAs on the y-axis, we have probably the strongest correlation that I've ever seen in anything that I've ever done, where we have what I'm guessing means a genomic co-occurrence of these elements that are happening at a very high rate and within the genomes of the asexuals. Now, before you ask me about this blue dot, I'm going to tell you that we have multiple lines of evidence to believe that this particular diploid lineage is actually an asexual. And we did not know this going into the project, and we've got all sorts of, it's pretty interesting that this, what we think is an asexual, but diploid is coming out clustering with other asexual lineages. So this is where we are with Kyle's project. We're right in the middle of things. We're really excited to get a better genome assembly that will let us figure out the extent to which we really do see co-occurrence of these elements in the Podemo genome in asexuals. We do have really clear evidence through the recent expansion of some repetitive elements, transposable elements, as well as these histones and rDNAs in the asexual lineages. What we're working on now is doing qPCR validation of the histone and rDNA results, as well as verification and analysis of the extent to which we see genomic co-occurrence of these elements within the genomes themselves. We're also doing, Kyle got one of the very last NSF DDIGs, which are now dead, to do transposable element evolution in what we call real time. He's using pool-seq to do population-level comparisons of transposable elements in sexuals and asexuals from the same natural populations to get a picture of what these transposable elements are doing on shorter timescales. So that's where Kyle's work is, and this is where I'm going to stop talking about harmful mutations, and I'm going to start talking about mutations, still mutations. They may or may not be harmful, but they're doing what looks like some interesting things in genes that are specific to sex and to maleness in our asexual snails. And so this is Joe Jelinski. He is co-advised by myself and John Logsdon, and he has been working on what it means to be a gene that is specific to sex or maleness in an asexual context. And in particular, our expectation is that genes and phenotypes that are used only for sex, so genes that are like only for meiosis, genes that are used to make sperm, and the sperm itself, should evolve differently in sexuals and asexuals. And so we're still thinking of this mutation accumulation framework. So here I have a rate of mutation accumulation on the x-axis. We can establish a baseline expectation for mutation accumulation in sexuals across the genome, as well as in sex-specific genes. Now, we expect that asexuals across the genome, because of this selective interference phenomenon, should experience a higher rate of accumulation of mutations, especially harmful ones in sexuals. If we look at sex-specific genes, we might expect this rate of mutation accumulation to be even higher, because now mutations that would be harmful in sexuals up here are neutral in an asexual context. So we're addressing whether we see the predictions laid out by this framework, are they met in our snails? And we can use this sort of analysis to illuminate consequences of asexuality as well as changes in selective regimes from purifying selection, maintaining a particular phenotype, to relaxed selection once reproductive mode is changed. So I'm just going to give you a quick snapshot of what Joe is doing with respect to molecular evolution. He's addressing, he's looking at patterns of molecular evolution in something like 80 genes associated with meiosis in sperm. This is just one example from the PRDM9 zinc finger array. This is associated with recombination hotspots in animals. And Joe has shown, just in a nutshell, that this particular array does evolve more rapidly in asexuals relative to sexuals. This is a figure that shows non-synonymous mutation accumulation with respect to theta, here scaled by the synonymous theta, so we're controlling for mutation rate differences. And what we find is that asexuals do have a higher rate of accumulation of these mutations in this particular gene associated with recombination in asexuals. And Joe's shown a number of other really intriguing patterns in genes associated with meiosis as well. I'm not going to talk much more about those results, though, because I wanted to talk about sperm. So Joe is actually looking at phenotypes specific to sex and how they're evolving in sexuals and asexuals. And what Joe is doing is he's taking advantage of the fact that our asexual snails, I told you that they're almost always female, but about 1 to 2% of the time the asexual females are making male babies. And we don't know what this is all about. We think it's probably some sort of error in oogenesis associated with aneuploidy. Whatever the mechanism, it does result in the production of males. We know that these males produce sperm. We also know that these males can engage in copulation. We don't know whether the copulatory attempts actually result in fertilization of females. But either way, we can use these males because they make sperm to compare sperm phenotypes in sexual and asexual lineages. So what Joe is doing, and this is a picture of a potamosperm. This is a normal potamosperm. I'm going to show you some fun pictures of some abnormal potamosperm. And Joe is getting sperm out of these males, and it's not fun for anybody. The males have to be dissected and so on. You know, there's nothing going on there that I can't talk about here. But the males are dissected. We take out the sperm, and then Joe measures. I can tell you how many he measures and how he figured that out if you'd like. But he measures about 12 sperm from each male, and he takes measurements from the head. So these are standard sperm measurements. He does a sort of qualitative, are they normal versus abnormal, overall shape, and then he does some quantitative measurements in image day. He does the same thing with the tail. And then what Joe does is he then thinks about these data, again, in the framework that I've been talking about all along. We think about how we might expect sex to influence particular traits or particular patterns of evolution. Here I've got a sperm trait on the x-axis, or y-axis, relative to, we've got here, I've got the outgroup. These are Potamopyrgus esterinus, and I'm going to now give you an additional nuance here. I've mentioned that we have a genome duplication in our snails, such that this Potamopyrgus esterinus, even though it is diploid, it has only about half the nuclear genome size of the diploid potamos. So diploid potamos, shown here in blue, they're diploid, or they seem to be diploid, but they've got about twice as much DNA in their nuclei as esterinus. The triploid asexual is 1.5 times the DNA content of the diploids, and the tetraploids, of course, have about twice the DNA content. Now, I'm showing this to you, these DNA amounts, because we're also now not just going to think about sex and ploidy, but we're going to think about one of the other key determinants of cell-level phenotypes, so sperm or cells, is nuclear DNA content. So we can make then expectations regarding how sex should influence sperm traits. So here we see if sex but not ploidy is influencing sperm traits, we should see differences, consistent differences in asexuals relative to sexuals. If ploidy is important, we should see consistent differences in asexuals relative to sexuals, but also the two ploidy levels of asexuals relative to one another. But if we want to also take nuclear DNA content into account, we can take advantage of the fact that esterinus has half the nuclear DNA content of diploid potamos. And then we can expect to see actually different sperm traits in sperm cells as we look across both sexual and potamos versus sexual esterinus, as well as the asexual potamos. So with this framework in mind, then Joe has measured something like 15 different sperm traits. I'm just showing you one outcome here. This is sperm head length on the y-axis. This result is remarkably consistent across pretty much all sperm traits that Joe measured in this quantitative fashion. So pretty much every time we see this pattern whereby Asterinus has the smallest value of the sperm trait, followed by diploid potamose, triploid potamose, and tetraploid potamose. So what this result suggests is that nuclear DNA content indeed seems to be the main driver of sperm morphology, though we also see a consistent pattern of significantly higher variance in sperm traits in asexuals versus sexuals. This is consistent with the expectation that we might have evolution under relaxed selection, potentially influencing sperm phenotypes in these asexuals. That particular conclusion is emphasized when we look at sperm ultrastructure abnormalities. So these are more qualitative comparisons of what these sperm look like. This is a sperm from one of these sexual Asterinus. It's what we think of as normal, though of course we have no idea really what normal sperm look like because we're the first people to study this. But Joe has actually characterized a number of quite fun and interesting phenotypes in sperm, ranging from the two-tailed through what he calls a bell. This macrocephaly phenotype is really interesting because sperm early in development have a giant head, and the giant head is supposed to go away. But Joe has seen some snails in which mature sperm still have this giant head. This is what he calls a teardrop, and these sperm have this weird kind of like elongated head that he calls jellyfish. So when Joe goes in and characterizes what these sperm look like, he finds that especially the triploid snails here in the middle on B, we look at the frequency of abnormal sperm. The triploids, about 80% of their sperm have one of these qualitatively abnormal phenotypes. Only 20% of the tetraploid male sperm is abnormal. A somewhat higher fraction of the diploid sexual sperm in potamose is abnormal than Asterinus, but we still see much lower frequencies of abnormalities altogether. The types of abnormalities are highlighted here in this figure, showing the type of sperm phenotype involved in the frequency of that sperm phenotype on the y-axis. Interestingly, the diploid sexuals, almost all the sperm phenotypes are abnormal and having two tails. What we see in the triploids and tetraploids is kind of the same signature of sperm abnormalities, where they seem to have a lot of these other phenotypes, and in particular, these things like head misshapen. This is head snap, where I think you're basically missing a head. The triploids almost look like a stretched out, higher frequency version of the tetraploids, really highlighting that these sperm that are produced by these asexually produced males are abnormal, and especially in a triploid context, which we think might point to challenges associated with having an odd number of chromosomes. So altogether, getting back to Joe's question of genes and phenotypes specific to sex and what they're doing, he does see that at least some sex-specific genes are evolving differently in asexuals and in a way that does look consistent with relaxed selection. Asexually produced males produce a relatively high frequency of abnormal sperm, and especially triploids. And Joe's really in the middle of doing more of this. So many more genes and phenotypes are on the way. So to put all this together where we are right now, I'm clearly in the middle of doing, I think, some interesting and hopefully exciting work that I think will cast some new perspectives on what it means to be sexual versus asexual in a natural population. So we do see good evidence for accelerated accumulation of likely harmful mutations in asexuals, ranging from radical amino acid changes through repetitive elements, and some mutations accumulating in sex-specific genes that could be harmful, they could be neutral, we don't know yet. We also have evidence for degradation of sex-specific phenotypes, in particular exemplified by these sperm produced by asexuals. We're doing a lot of other things. We're looking at things like mitochondrial function, evolution and coevolution of mitonuclear proteins in a sexual and asexual context, as well as adaptive molecular evolution. We are also using these genomic resources to do a lot more things, including evolution of invasive phenotypes, and we're even doing some work associated with ecotoxicology. These snails are a model for studying water quality, and we're starting to get into that area as well. So with that, I will wrap up. Lots and lots of people, every one of them, I hope, has pictures in this snail. And I want to say thank you to them and to my funding sources, primarily the National Science Foundation, and all of you for showing up.
I'm sure Maureen will take questions.
Yes?
So I have two questions.
Mm-hmm.
The first being, are there ecological markers to these lakes that predict polyploidy level or sexual reproduction mode? And then the second question is about the nuclear DNA content and whether you looked at that in other cell types to give links to other different things.
Yeah, no, those are good, really good questions. The first question, are there ecological differences between lakes with more or less sexuals and asexuals and different ploidy levels? So the work that Kurt Lively has done for the last few decades has demonstrated very nicely that there is a positive association between the frequency of infection by a castrating trematode worm and the frequency of sex, such that in many cases, but not all cases, lakes with a lot of sexuals also have a lot of disease. And he's shown really nice evidence for negative frequency-dependent selection exerted by these worms that favor sex. Now, that said, there are a lot of New Zealand lakes that have a lot of sex and really no infection. And the worm incidence itself only explains about 25% of the variation in the relative frequency of sex. So that's certainly an important ecological variable, but it's not the whole story. I have been really interested in addressing whether material costs associated with having extra genomes in cells, and in particular costs associated with needing to have a lot of phosphorus in the body if you are a polyploid individual. And we've actually shown that phosphorus content on a per gram of, or on a per unit of mass in these snails goes up as ploidy level goes up. If this might mean that asexual polyploid snails have higher phosphorus requirements than sexual diploids. And we have actually shown a ploidy effect in that the tetraploid snails need more phosphorus than the triploid snails do. We haven't yet been able to do the same experiment in diploid versus polyploid snails because the sexual snails are actually pretty hard to culture in lab. So the system can be very difficult. But I am in the middle of trying to get enough funding to really do a very comprehensive survey of nutrient availability in New Zealand lakes. We did kind of a preliminary survey, and that was promising, but we need a lot more money to go back and do it. And unfortunately, this isn't a good time to get a lot of money in the United States for science, but we're trying. The second question, are you asking about like endopolyploidy? It's a really interesting question. We have never done the kind of tissue-specific assessment of cell contents that we would need to do that. These snails are really small, and we have a hard time sometimes getting enough cells from a little piece of tissue to do that kind of thing, but I think that it is warranted. We certainly see what looks like, I mean, I wouldn't call it ploidy mosaicism, but we certainly see some variation in nuclear DNA contents with if we grind up a whole snail. Like one really fun thing that actually led us to do some of this work with males is that if you grind up a whole male snail, you get a peak associated with what's going on in their gonads. And the diploid males have a very nice, like pretty tight haploid peak associated with sperm. And the polyploid males have a very broad, not haploid peak, but a not quite somatic ploidy peak either that we think is associated with very high levels of aneuploidy in the sperm that they're making. But I think it's something that we would like to do more of. I wish they were larger, you know.
Before we take one more question, just two quick things. Ken has a ride share going to pizza tonight, so you can talk to him. And then secondly, just a reminder that graduate postdoc, there's a sushi lunch on the second.
Ooh.
After this.
Sushi. Wow. I was expecting pizza. That's awesome.
Pizza tonight.
Yeah.
Sushi for lunch.
But that's
just
no, that's great. That's awesome. All right.
Maurine, have you looked at the age distribution? Oh, by the way, I'm very pleased that the influence has actually had an effect over time. But have you looked at the age distribution of TV sexual versus asexual? What you probably would see in the sexual is very rapid turnover of TV.
Mm-hmm.
Why it's hard to be consensual at the start.
Yeah.
And whereas in the asexual, you should see much more uniform age distribution.
Yeah, we're in the process of, like, we need better genome assemblies from the resequencing data to do that. One super interesting, I was just looking at I don't think I have the slide here. Do you guys know what a TE landscape looks like? No? I wonder if I can, can I open something? Okay, give me 1 second. This is really interesting. Oh no, I have to go, this is, never mind, I'm not going to do it. So there's something called a TE landscape that looks like a histogram. And so what you do is you plot divergence of TEs from one another in the same TE family on the x-axis and frequency on the y-axis. So you have a histogram showing the frequency distribution of essentially evolution within TE families. And so I think to get to Lauren's question, the further out along that axis you are, the older the TE family is. And if we compare the distribution of TE age in potamos relative to Estuarinus, potamos have this like huge burst of new TE activity relative to Estuarinus, and that's in our sexual diploid line. Once we are able to do reference-guided genome assemblies with our resequencing data, like once our de novo draft assembly is good enough to do that, we're going to do exactly that. But I think that you're right that could be really interesting, and I think that if we see those sorts of old versus new patterns in sexuals versus asexuals, we can learn a lot. You're right, if they have, depending on what's going on with the TEs, right? No, that's an interesting point. Maybe we should have, we started with a sexual diploid because we thought that would be easier. But if you're right, if we have like crazy TE stuff happening, especially in the context of a degenerate tetraploid and all that kind of stuff, it could be, maybe our lives would have like been way easier if we just would have used a triploid. But it's too late. It's way too late for that. But well, that's a really interesting suggestion. Yeah.
So in your trees, you showed that asexuality evolved once. Have you found any instances or any times or where it's evolved multiple times?
Yeah, it's actually when we do population genetic kinds of analyses, like we've done a bunch of nuclear SNP genotyping of snails within the same lakes, of sexuals and asexuals. We see that asexuals are more closely related to sexuals in their same lake than they are to asexuals in other lakes. And we also see a lot of genotypic diversity in asexuals even within lakes that doesn't look like just mutational divergence. So what we think is happening is that for some, like, we have completely no idea why this happens in the sexual snails to such a great extent, but we think that sexual snails have some sort of propensity for making asexual babies in the system. We wonder if it's linked, it could be linked to the genome duplication. The other snails, like close relatives don't do this. Our snails also have internal brooding of offspring, and other snails that are related to them tend to lay eggs. So we also wonder if maybe there's something about the life histories of these snails that predispose them towards asexuality. But for whatever reason, the sexual snails again and again are making asexual descendants. And then the transition seems to be one way and irreversible.
I think we'll...
Michelle.
Michelle asked a question because it was her birthday yesterday.
So I thought it was funny that the snail, the facelessness of the snail directly parallels Kurt's career and your career. And now you find it on the west coast of Vancouver Island. I was wondering if you've been there recently, and do you have any, more seriously, if you have any thoughts on whether invasion is going to be like selection?
That's a really good question. I didn't know it was on the west coast of Vancouver. Did you say Vancouver Island? Oh, I would like to go there. That sounds like a good reason. No, we've been horrified at the idea that we might be associated with spreading these snails around, but I don't think so. They haven't been found in Iowa yet, except in a pet store in Iowa City where I discovered them, and I called the DNR on the pet store. So I was a good citizen, but they didn't come from me. But the invaders, and they're invasive in every continent now except for Antarctica, they're always asexual. We don't know why. It could be a chance kind of thing. It could be that the populations that have been introduced have been asexual, because a lot of populations are all asexual. And it could be a selection sort of process. And I have a master's student who's working on that kind of indirectly. He's looking at the genetic and phenotypic variation of some of these invasive populations in North America. We don't know. I mean, obviously there's a colonization advantage if you're asexual. There could certainly be distinct advantages associated with maintaining a well-adapted multi-locus genotype. There could be associated advantages with being polyploid that aren't really about being asexual. So we don't know. I don't think that it's hard to study because you can't really say to like the U.S. or presumably the Canadian government, can I take some snails and put them in a place where they're not already and see what happens? And I don't think that I would be allowed to do that. And because the sexual snails haven't invaded, I think it's been pretty hard to do the kind of like powerful, even comparative experiments that you could do to learn about that. But they seem to be, they're doing pretty well.
So one last thing I'll say is that there is a little bit of an opening at the end of the day. So if you've got a set of questions you really want to ask, let me know. please join me in